Welcome everyone. I'm excited to kick off our presentation today. The topic is the electron transport chain and ATP synthesis, a fundamental process in biochemistry. On this first slide, we're just setting the stage. Over the next few slides, we'll explore how cells produce energy, step by step. So let's dive in and get started. In eukaryotic cells, the processes of electron transport and oxidative phosphorylation take place across the inner mitochondrial membrane. These are crucial steps in cellular respiration because they help us reoxidize NADH and FADH2, which are generated in various metabolic pathways like the citric acid cycle, glycolysis, and fatty acid oxidation. Specifically, NADH and FADH2 are produced in the mitochondrial matrix during the citric acid cycle and fatty acid oxidation, while glycolysis occurs in the cytoplasm. The key point here is that these molecules need to be reoxidized to continue fueling energy production. As electrons are transferred through the electron transport chain, energy is released and ultimately used to produce ATP, the energy currency of the cell. This process, called oxidative phosphorylation, is actually the major source of ATP in our cells. Now, in prokaryotes, the setup is a bit different. Since they lack mitochondria, these electron transport and oxidative phosphorylation components are embedded in the plasma membrane. This highlights how these fundamental energy production processes are conserved, but adapted to different cellular architectures. Overall, these processes are central to how cells generate the energy they need for all their functions, linking metabolism directly to energy production across different organisms. Let's talk about redox potential. First, oxidation is all about the loss of electrons. When a molecule undergoes oxidation, it gives away electrons to another substance. On the flip side, reduction involves gaining electrons. So, when a molecule gets reduced, it gains those electrons. Now, in any chemical reaction involving oxidation and reduction, things are interconnected. If one molecule is oxidized, that is, it loses electrons, then another molecule must be reduced by gaining those electrons. This transfer of electrons is what we call an oxidation reduction, or redox, reaction. Understanding this transfer is crucial because it explains how energy is transferred in biological systems, like in cellular respiration or photosynthesis. So always remember, oxidation and reduction go hand in hand. One cannot happen without the other. When NADH donates its electrons, it gets oxidized to NAD+. This process is crucial because it allows NADH to act as an electron carrier, transferring energy from one part of the cell to another. Essentially, NADH loses electrons during this step. On the other side, molecular oxygen acts as the final electron acceptor in the chain. When it gains those electrons, it's reduced to water. This gain of electrons by oxygen is what drives the production of water and helps complete the electron transport chain. These processes are fundamental to cellular respiration, as they enable the cell to efficiently produce energy in the form of ATP. So, in summary, NADH loses electrons to become NAD+, and oxygen gains those electrons to form water. Both are key steps in energy transfer within the cell. Let's dive into the concept of oxidation reduction potential, or E, which is also known as the redox potential. This is essentially a measure of how much a substance wants to gain or lose electrons. Think of it as the substance's affinity for electrons. And importantly, we measure this potential relative to hydrogen, which serves as our standard reference point. Now, when we talk about a positive redox potential, that indicates that the substance has a higher affinity for electrons than hydrogen does. In other words, it's more eager to accept electrons. So, such a substance would be willing to accept electrons from hydrogen, which is a key concept in understanding redox reactions. For example, oxygen is a classic case of a strong oxidizing agent. It has a high redox potential, meaning it readily accepts electrons. This property makes oxygen crucial in many biological and chemical processes where oxidation occurs. Overall, understanding the redox potential helps us grasp how different substances interact during redox reactions, which are fundamental to energy transfer in living organisms and various industrial processes. Now, let's talk about negative redox potential. When a substance has a lower affinity for electrons than hydrogen does, it means that it's more likely to donate electrons rather than accept them. So, in this case, the substance would give up electrons to hydrogen ions, or H+. This process effectively reduces the H+ ions to form hydrogen gas. An example of a substance with a strong tendency to donate electrons is NADH. NADH acts as a powerful reducing agent because it readily donates electrons during metabolic reactions, helping to drive many biological processes forward. This concept of redox potential is essential because it helps us understand how electrons are transferred in cellular respiration and energy production. When we look at biological systems, one of the key concepts is the standard redox potential for a substance, which is denoted as E0. This value is specifically measured at a pH of 7, which is physiologically relevant, and it's expressed in volts. Now, in oxidation-reduction reactions, or redox reactions, electron transfer is happening. This is the core process. Electrons move from one molecule to another, driving many biochemical processes. The total voltage change for the entire reaction, which we call delta, reflects the overall change in electric potential. Think of it as the sum of the voltage changes that occur during each individual oxidation reduction step within the reaction pathway. So, each step has its own small voltage change, and when you add them all up, you get the total voltage change for the entire process. Understanding these voltage changes helps us quantify how likely a reaction is to occur and how energy is transferred within the cell. Essentially, these concepts are fundamental because they connect the electron transfer events to the energy flow that powers biological functions. Now, let's take a closer look at the standard free energy change of a reaction at pH 7, denoted as delta 0. This value can be calculated based on the change in redox potential, which is represented as delta 0. Essentially, delta 0 tells us how much free energy is available or required during a redox reaction under physiological conditions. The key formula here is delta 0 equals negative N times F times delta 0. In this equation, N is the number of electrons transferred in the reaction, which is crucial because more electrons mean a larger energy change. Delta 0 is the difference in redox potential between substrates and products, measured in volts. The delta 0 value is expressed in kilocalories per mole, giving us a sense of the energy magnitude involved. And F is a constant called Faraday's constant, which is 23.06 kcal per volt per mole. This constant helps convert the electrical potential difference into energy terms. Understanding this relationship is fundamental because it links the electrochemical properties of molecules to the energy available during metabolic processes. It helps us see how changes in redox potential directly influence the energetics of biochemical reactions, which ties back to the overall theme of energy transfer in biological systems. A reaction with a positive delta E0 prime has a negative delta G0 prime, meaning it is exergonic. Essentially, when the standard reduction potential increases, the free energy change becomes more negative. This indicates that the reaction tends to proceed spontaneously. So, for this reaction, the more positive the E0 prime, the more energy is released, making it energetically favorable. Remember, this is a key concept in understanding how redox reactions drive biological processes. It ties into the overall theme of how electrons transfer and energy flow within cells. Now, let's take a closer look at how electron transport occurs from NADH. As you know, NADH is a key electron donor in this process. It donates electrons to the electron transport chain, specifically to complex I, also known as NADH dehydrogenase. When NADH hands over its electrons, it gets oxidized back to NAD+, which can then be reused in metabolic pathways like glycolysis and the citric acid cycle. These electrons then travel through a series of protein complexes embedded in the mitochondrial membrane. As they move along this chain, they release energy, which is harnessed to pump protons from the mitochondrial matrix into the intermembrane space. This creates a proton gradient, essentially a stored form of energy that powers ATP synthesis. This process from NADH is crucial because it contributes significantly to the overall production of ATP, fueling many of the cell's activities. Remember, understanding how electrons move from NADH helps us appreciate how our cells efficiently convert nutrients into usable energy. So, when we look at NADH oxidation and ATP synthesis, it's important to understand that these processes do not happen in just one step. Instead, they involve multiple stages that work together efficiently. Now, a key point to remember is that electrons are not transferred directly from NADH straight to oxygen. Instead, there's a series of transfers involved. Electrons move from NADH to oxygen, but they do so along a chain of specialized molecules called electron carriers. This chain is known as the Electron Transport Chain, or sometimes the respiratory chain. It acts like a relay race, passing electrons from one carrier to the next, ultimately leading to oxygen being reduced to water. Understanding this chain is crucial because it explains how energy from NADH is gradually used to generate ATP, rather than all at once. This stepwise process helps us appreciate the efficiency and regulation of cellular respiration, which is fundamental to how our cells produce energy in a controlled and sustainable way. Let's take a closer look at the organization of the electron transport chain complexes. As you can see, these complexes are arranged in a specific order within the inner mitochondrial membrane. Complex I, also known as NADH dehydrogenase, is the first step where electrons from NADH enter the chain. Moving along, we have complex II, or succinate dehydrogenase, which feeds electrons into the chain from FADH2. Next, electrons are transferred to complex III, the cytochrome BC1 complex, which plays a key role in further passing electrons down the chain. Following that, we have complex IV, the cytochrome C oxidase, where the electrons are finally used to reduce oxygen to water. Understanding this organization is crucial because it highlights how electron flow is tightly coordinated, enabling efficient energy production. This arrangement also facilitates the creation of a proton gradient across the inner mitochondrial membrane, which is fundamental for ATP synthesis. Overall, the organization of these complexes is central to how mitochondria generate the energy that powers virtually all cellular activities. Let's take a closer look at the electron transport chain. This process involves three large protein complexes that are embedded in the inner mitochondrial membrane. These complexes work together to facilitate the transfer of electrons and ultimately produce ATP. The first one is NADH dehydrogenase complex, also known as complex I. It receives electrons from NADH and begins the chain. Next, we have the Succinate Q reductase, which is involved in transferring electrons from succinate. Then, there's the cytochrome BC1 complex, also called complex III, which further moves electrons along the chain. Finally, cytochrome oxidase, or complex IV, completes the process by transferring electrons to oxygen, forming water. These complexes are key players because they create the proton gradient used to generate ATP, which is essential for cellular energy. So, understanding how these complexes work together is fundamental to grasping how our cells produce energy efficiently. Electrons flow from NADH to oxygen through these three complexes, which are the key players in the electron transport chain. Each of these complexes contains several electron carriers that work sequentially, meaning they pass electrons down the chain step by step. This orderly transfer is crucial for efficient energy production. Now, these large complexes don't work alone. They need two additional free electron carriers to connect everything smoothly. The first is ubiquinone, also known as Coenzyme Q or CoQ. It acts as a kind of shuttle carrying electrons between complexes. The second is Cytochrome C, which transfers electrons from complex III to complex IV. These small but vital carriers are essential because they facilitate the flow of electrons, ensuring the entire process runs efficiently. So, in summary, electrons travel from NADH to oxygen, passing through these complexes and their carriers, with CoQ and cytochrome C serving as crucial links in this chain. This flow is the core of how our cells generate the energy they need to function. Now, let's talk about ATP synthesis during oxidative phosphorylation. Essentially, NADH and FADH2, which we've discussed earlier as key energy carriers, are oxidized by the electron transport chain. This process involves the transfer of electrons through a series of complexes, ultimately leading to the synthesis of ATP. What's really important here is the energy released during this electron transport. This energy is harnessed to create a proton gradient across the inner mitochondrial membrane. Think of it like a reservoir of potential energy. This gradient is crucial for the next step. This is where the chemiosmotic hypothesis comes into play. The proton gradient drives protons back across the membrane through an enzyme called ATP synthase. As protons flow through ATP synthase, it catalyzes the formation of ATP from AD and inorganic phosphate. So, in essence, the energy from electron transport is converted into a proton motive force, which then powers the synthesis of ATP. This process is fundamental to how our cells generate the energy they need to function efficiently. So, what we're seeing here is that the proton gradient is really the key link between electron transport and ATP synthesis. It's what drives the production of ATP, but it's important to note that this process is different from substrate-level phosphorylation, which involves a direct chemical transfer. Instead, here, the energy from electron transport creates a proton gradient across the inner mitochondrial membrane. This gradient then acts like a battery, providing the energy needed for ATP synthase to produce ATP. Now, about the enzyme involved, originally called it PACE, it's a bit of a misnomer. Because without the input of energy from electron transport, this enzyme can actually run in reverse and hydrolyze ATP instead of making it. So, in essence, the direction of this enzyme's activity depends on the energy available, whether it's synthesizing ATP using the proton gradient or breaking it down when that gradient isn't present. This highlights how tightly coupled the process is, and it's a perfect example of how cellular energy dynamics are finely balanced to meet the cell's needs. Let's review the key points from this slide. Electron transport occurs along the respiratory chain, starting with NADH oxidation. As NADH is oxidized, it drives the pumping of H+ ions across the inner mitochondrial membrane. This pumping is carried out by three main complexes: NADH dehydrogenase, the cytochrome BC1 complex, and cytochrome oxidase. Each of these acts as a H+ pump, moving protons from the mitochondrial matrix into the intermembrane space. Now, an important concept here is the free energy change involved in transporting these charged ions across the membrane. Because H+ ions are charged, their movement isn't just about chemical gradients, but also electrical ones. This creates an electrochemical proton gradient, essentially a stored form of energy that the cell can later harness to produce ATP. So, in summary, electron transport leads to the pumping of H+ ions, setting up this vital electrochemical gradient, which is fundamental for the cell's energy production. This process illustrates how electrons transfer energy into a form that the cell can use efficiently. Alright, so let's break down what's happening during this stage of cellular respiration. The process starts with the pumping out of H+ ions, which creates a higher concentration of these protons in the intermembrane space of the mitochondria. This buildup results in two key things. First, it establishes a concentration gradient, but second, it also creates an electrical potential across the inner mitochondrial membrane. This electrical potential makes the side facing the intermembrane space positively charged. Because of this electrochemical gradient, think of it as a stored form of energy. Protons are inclined to flow back into the mitochondrial matrix. They do this through a special enzyme called ATP synthase. Now, this flow of protons back across the membrane is actually what drives the synthesis of ATP, which is our cell's main energy currency. The entire process is powered by what we call the proton motive force. This force isn't just one thing. It's made up of two components: the pH gradient, which relates to the chemical difference in H+ ion concentration, and the membrane potential, which is the electrical charge difference across the membrane. Together, these components create a powerful energy source that enables ATP synthase to produce ATP efficiently. Understanding this helps us see how the mitochondria convert electron transport into usable energy, an essential part of cellular respiration. So, when FADH2 is reoxidized, it happens through a process involving ubiquinone, which acts as an electron carrier. This oxidation process causes H+ ions to be pumped out of the mitochondrial matrix, but importantly, only by the cytochrome BC1 complex and cytochrome oxidase. This is a key point because it explains why the amount of ATP generated from FADH2 is less compared to NADH. In fact, measurements show that for each NADH molecule that gets oxidized, about 2.5 ATP molecules are produced. In contrast, only around 1.5 ATP molecules are generated from each FADH2 molecule. This difference highlights how the specific entry point of electrons into the electron transport chain influences the overall efficiency of ATP synthesis. It ties back to the overall theme of how different molecules contribute differently to our cell's energy production. Let's wrap up with a quick summary of electron flow. As we've seen throughout this presentation, understanding how electrons move is fundamental to grasping how electrical circuits and devices work. The key points to remember are that electrons flow from areas of higher to lower potential, creating the current we rely on daily. This flow is influenced by factors like voltage, resistance, and the material properties of conductors. Recognizing the direction of electron flow helps us better understand circuit behavior and troubleshooting. Overall, mastering these concepts connects directly to the theme of controlling and harnessing electrical energy effectively. Thank you for your attention, and I hope this summary solidifies your understanding of electron flow.